This is a very, very special night for us for two reasons. First, of course, the reason most of you are here is we have the privilege of hearing two world-renowned science writers, David Quammen and Zimmer, and we'll hear more about them in a minute. But there's another reason, and that is that we are celebrating the 10th anniversary of the Evolution Matters series, which is our flagship lecture series for HMNH. And I can say, because I was looking, over the last 10 years, this lecture series has featured distinguished evolutionary biologists, both from our own Harvard faculty and colleagues from outside the university. Topics have ranged across the tree of life, from E. coli to Darwin's finches to the humble, or according to the speaker, not so humble, mushroom. From the earliest history of Earth to rapid evolution happening in our own lifetimes. Talks have ranged from finding your inner fish to the amazingly titled Paleo Fantasy, what evolution really tells us about sex, diet, and the way we live, as well as taking us around the world from island biodiversity in the Caribbean, fishes in the Congo, and humans in Framingham, Massachusetts. And of course, tens of thousands of people have watched our lecture videos as well, so people from around the world. Since the very beginning, Evolution Matters has been generously supported by two of the museum's greatest friends, Drs. Herman and Joan Suit. And they've given me permission to say this. This evening, I'm thrilled to announce that they have made a major gift to endow the lecture series, thereby securing its future. And Herman, Joan, we wouldn't be able to do this without you. And I'm so grateful that you share our passion for education and providing opportunities for people to learn so much more about this subject matter that is fundamental to our understanding of life on Earth. Thank you. But it's now my pleasure to introduce a dear friend, Janet Browne, Aramont Professor of the History of Science, who will be introducing our speakers and moderate the Q&A later. Janet was actually our very first Evolution Matters speaker in February 2009 when she gave a talk, Darwin at 200, Rethinking the Revolution. As many of you know, she has specialized in reassessing Charles Darwin's work, first as associate editor of the early volumes of the Correspondence of Charles Darwin, and more recently as author of an award-winning 2-volume biographical study that I'm sure, as me, many of you have in your libraries, and that integrated Darwin's science with his life and times. There was no one better to give our very first lecture, and no one better to introduce our speakers. Please join me in welcoming her to the podium. Thank you.
It's a real pleasure for me to be able to welcome you, as we've already been welcomed by Jane, but to also say it again from those of us who have an interest in the history of natural history and natural history in its own right. It's such a delight that we have two such dynamic and eloquent writers to speak to us today. Most of us will know our guests through their pages, but it's a real pleasure that we actually have them here to talk to each other and to us about their work. That is actually how I have come to know both of our speakers, is first through their pages. And tonight is special for me because it's the first time I've actually met Carl in the flesh, which is delightful. But I do know David Quammen from before, and might perhaps just forego all the formal structure about the introduction and to say that we met, David and I, with a number of other people, at a very forbidding weekend that he perhaps will remember as well as I do, where we were all meant to chat about the craft of science writing and be recorded and then published. But it was never published. So I'm assuming, perhaps with David, that we didn't have the right kind of chat. which I'm sure we'll get tonight. So that's fine. And I, of course, know Carl through his illuminating books on evolutionary theory. We're very fortunate to have two such gifted writers with us tonight. What I would like to say is that both of our speakers have had a long and distinguished career, and I understand that they began their intellectual lives as contemporaries at a university further south from here in New Haven. And it may be that we'll hear a little bit about that as we go through the evening. And both of them took to science writing through early careers in newspaper writing, journals, periodicals, and have then gone onwards to become, I think, the most widely regarded writers on scientific matters to do with natural history, I would say in the world, certainly in North America. David Quammen and Carl Zimmer both have new books out, which I hope we will hear about this evening. David perhaps will be best known to you through The Song of the Dodo, many other books that he's written, but that is a very evocative text that most of us, many of us, maybe all of us, have read. And Carl has a long career writing on a number of different topics. Evolution is only one of his topics, but that is the one which I know best. I would like to mention that he's the only science writer, I don't know if this is, yeah, you claim it on your website. It's the only science writer to have a species of tapeworm named after him. And that's a pretty rarefied category. But in this audience, you know. But you will know his delightful book, The Tangled Bank, An Introduction to Evolution, and then Evolution, The Triumph of an Idea. So your two speakers and myself and many of us here are united through a real appreciation of evolutionary theory. For myself, for the role of Charles Darwin in that achievement, but I think we are all very fortunate indeed to have two such gifted writers with us tonight to talk about their work and their love of natural history. So please, would you join me?
It's great to be here. And we don't even know who's going to start and who's going to ask the other person the first question, but I'm going to exercise rank of age and ask Carl to let me say a couple of words to start. In addition to thank you, Janet, for that eloquent introduction, I'm going to emend one small part of it. And that is, you outed us a little bit and said that we were at a certain university in Connecticut, south of here, which is true. Carl and I were both at that university, English majors, but we were not there as contemporaries. I went to New Haven the year Carl was born, I think.
Yes.
So I just wanted to straighten that part out, in case it wasn't obvious already.
I was born in Yale New Haven Hospital.
You were? Oh,
yeah.
Okay.
So we were both there.
So we both arrived in New Haven in 1966. Yeah.
Yeah, we should have hung out, you know.
Yeah. Yeah. So who first?
Well, I would love to start. I mean, we both brought each other's books with notes in them, and I have a bunch of questions I want to ask you, and you can ask me. So
I do, and partly about your book, partly about other work, and partly about some large ideas in terms of the treatment of science and the treatment of evolution.
So let me just jump in. The thing is that this book was so interesting to me because for a long time, I will talk to biologists and they'll say, you know, there's somebody who should have won the Nobel Prize, and they all say it's this guy named Carl Woese. And I have come to agree with that, I've come to appreciate that, but I still don't think that people really know who he is. And it's great that through this you have brought this invisible giant to attention.
Carl Woese, yeah.
The book is called The Tangled Tree: A Radical New History of Life.
Thank you.
Highly recommend. Yes, please get it. It's wonderful, and it's about the tree of life, but it's about Carl Woese. And I just wanted to read a couple quotes. I've always admired your ability to just like pluck out things that people say and then drop them into your pieces. So we're not hearing people going on for paragraphs and paragraphs, but just a sentence is enough to get who they are. And there are these two like almost understated quotes that sort of capture his career. One is where in the 60s he said, he said, a slight diversion in my research program would be necessary, where he was trying to figure out the genetic code and he realized there was something else he had to do. And then there's just one other little quote.
Trying to figure out the evolutionary origin of the genetic code.
Right, yeah. And then there's this other quote here, almost like bookends it. Then it dawned on me there was something out there, where he's discovering archaea. I mean, maybe you could sort of explain, like, what was it? What was his diversion?
Okay.
And what was out there? I mean, just so that people can appreciate what Woese did.
This guy, he's the central character of the book. You're right. Carl R. Woese, W-O-E-S-E. I try not, when I meet somebody in an elevator, to say that I've just published a book that's the history of molecular phylogenetics. I find that's not a very good elevator pitch. So what I usually say is I've just published a book about the most important biologist of the 20th century that you've never heard of. And then they say, oh, who's that? And then I say, Carl Woese. I said, never heard of him. My point.
Confirmed.
Carl Woese was a microbiologist at the University of Illinois in Champaign-Urbana from the mid-60s into the 21st century, who, as Carl said, conceived a deep urge to understand the earliest evolutionary history of planet Earth, the deepest branches on the tree of life, how things were related to one another right at the beginning, 3.5, 3.8, 4 billion years ago. And in order to do that, he invented a new methodology, a new technique that we now call molecular phylogenetics, taking long molecules, DNA molecules, RNA molecules, potentially proteins. He actually picked up a suggestion from Francis Crick, who said at one point that perhaps, the sequences in long molecules such as proteins would tell us a lot about the history of life. Woese picked up on that and decided, I'm going to try and discern the deepest shape of the tree of life by extracting a particular molecule that exists in all cellular life on planet Earth, one molecule. And then I'm going to sequence it, and I'm going to compare those sequences to see how things are related. And in the course of doing that, he sequenced this molecule in some microbes that everybody had taken for 300 years to be bacteria. And what he found by sequencing this molecule in those microbes was that not only were these things that looked like bacteria and acted like bacteria and walked like bacteria under a microscope, he found they were not only not bacteria, but they're more closely related to us than they are to bacteria. They're more closely related to eukaryotes, all organisms composed of complex cells, than they are to bacteria. And they ended up being named archaea. And he hit the front page of the New York Times above the fold on November 3rd, 1977 for that discovery. And it gave him his 15 minutes of Andy Warholian fame. But it's not the most important thing that he did, or the most interesting thing, or the most influential thing. So the book is all about him, but that's the starting bite on Carl Woese.
So you have this molecular tree which, you know, I mean, it looks different than sort of the old trees of life, and that you've got archaea and things. You're sort of moving the branches around, but you know, as your title indicates,
Yeah.
you know, the tree starts to get tangled. It
Yeah.
starts to get more interesting.
Yes, and that was why Woese was really important, is because other scientists picked up on his methodology in finding this particular molecule in different forms of life, sequencing it, comparing it. That turned into this discipline, molecular phylogenetics, using long molecules to discern who was how closely related to who else. Excuse me. And what scientists, Woese and others, started finding was that the tree of life was really not tree-shaped, or not at all perfectly tree-shaped. Was it a bush? No. Was it a web? No, not exactly. It was tangled, hence my title. Tangled by the fact that one of the most basic things about a tree shape is that it starts from one trunk, it rises through time, branches, it diverges into limbs, limbs diverge into branches, branches diverge into twigs. You have a canopy of these little tiny twigs, and it's all about divergence. What Woese and the people who followed him discovered was that there was also convergence of lineages, limbs and branches that flowed into one another, which is supposed to be impossible, and it is impossible in a natural tree. But it has been happening for 3.8 billion years in the history of life. And one of the labels for this phenomenon is horizontal gene transfer. Horizontal gene transfer. And that was what started me on this. I started reading, I might have been reading some of your pieces or Ed Yong's pieces, journalistic pieces in early 2013. And I first heard about horizontal gene transfer, and then I started reading some more about it, and then I discovered a fellow named Ford Doolittle who worked on it, and I discovered this fellow Carl Woese. And essentially I went down the horizontal gene transfer rabbit hole for 5 years.
For 5 years. So what keeps you down there in the rabbit hole? I mean, well, for you, what is so like absorbing about it? I mean, you end up with a great book, but I'm just wondering what.
Well, it got more and more complicated. It got curiouser and curiouser. I started talking. Woese was already dead. I never met Carl Woese. I started talking to the people who had worked with him, the people who knew him, other people in the field like Ford Doolittle. I took on a new understanding of Lynn Margulis's work on an idea called endosymbiosis, which is the idea now known to be factual, that some of the crucial internal organs in the cells of all of us complex creatures are the descendants of captured bacteria. The mitochondria that are the energy-producing organelles in our cells are descended from captured bacteria. The chloroplasts in plants that photosynthesize are descended from captured bacteria. That's the idea of endosymbiosis. People thought Lynn Margulis was nuts when she proposed this, but then it was proven, it was confirmed by molecular phylogenetics, by these molecular techniques. All of that and more is what kept me in the rabbit hole for 5 years. I thought I knew 3 good reasons why horizontal gene transfer was impossible when I first read about it. And one of them being the Weissman barrier, August Weissman, one of your, a minor character in there, a little bit more important in here because you really went into August Weissman. But it turns out that it is possible, it is real, it is important and consequential, and it's very counterintuitive. Okay, now I get to ask you something.
Sure.
I want to ask you eventually about some technique things, particularly one thing you do at the beginning. Well, maybe I'll ask about this right now. You begin with Luther Burbank, or at least near the beginning. Carl brings in the character Luther Burbank, the most famous plant breeder in America in the early 20th century. I assume that Burbank, California is named for him, but I don't know that for sure. No? Okay. Anyway, he was in Northern California.
But the Burbank potato is.
Okay. So here is this character that most of us have heard of but know nothing about. Luther Burbank, an intuitive plant breeder who was admired around the world for the variance of different important commercial and sustenance crops that he produced, including potatoes, tomatoes, I'm sure, a number of other things. One of the, and I don't have it, I think, marked to quote from it or ask you to read it, but very early in the book, Carl describes a day in which Luther Burbank, at his little place in Northern California, gets a visitor, Hugo DeVries, one of the rediscoverers in 1900 or 1901 of Mendelian genetics. Arguably the leading geneticist in the world in 1900, 1901, goes to visit Luther Burbank. And that seems to me not just a really nifty scene that leads to some surprises, but a brilliant synecdoche, something that is real and yet stands for more than itself. Because Luther Burbank doesn't go visit DeVries. DeVries comes to Luther Burbank. Tell us a little bit about how you chose to arrange that, how you chose to focus on that little episode, and what DeVries discovered.
So I decided to write a book about heredity, and I was determined to tell stories about heredity that people would enjoy reading because they hadn't read them a million times since 8th grade. So Mendel gets put way down. So I give Mendel the credit, but I don't say like, oh, by the way, maybe you haven't heard of this guy named Mendel. Of course, everyone's heard of Mendel. But Mendel's not the only person. It's a long history. Mendel built on what other people were doing. Mendel's stuff kind of disappeared because people weren't really prepared to make sense of it. And it was in 1900 when Hugo DeVries and other people start to rediscover Mendel, but they've been doing all their own research on cells, on chromosomes, their own breeding experiments, and they're like, wait, I think his magic 3-to-1 ratio, it tells us something important. But the thing is that heredity was not just a series of scientists just working in isolation and having these abstract thoughts. Heredity has just been profoundly important to humans forever, as long as there have been humans. And one of the ways in which it's important is through breeding. We inadvertently bred domesticated crops, and then in the 1700s, people started to say like maybe we could control this. We have all these breeds. How did this happen? Maybe there are rules to it. And Mendel was one of many people who was trying to figure that out. And Luther Burbank was this sort of American sort of folk version of that. And there were a lot of people like Luther Burbank in America, but he was the most successful in that he had this, they called him the newspapers would call him the plant wizard, because somehow he would see the potential in the heredity of these plants for something fundamentally new. And so you would suddenly have the Burbank potato, which is way better than the potatoes that came before, or the Shasta daisy, or a spineless cactus. I mean, it was just like one thing after another that he would concoct. And it was one thing for Hugo DeVries and other people to say, okay, we understand the rudiments of what Mendel was trying to get at, and we think it has something to do with chromosomes, but still, even with the dawn of genetics, they knew so little about heredity, and they thought, Burbank must know. And so all these scientists would want to be pals with Luther Burbank. Like, maybe we could kind of get on the inside with him and see what he does. And so Hugo DeVries went all the way from the Netherlands to California. He was supposed to officially give a lecture at Stanford, but he wanted to go see Burbank. And he hung out with Burbank, and he ate these stoneless plums, and he's like, this is amazing, and how does he do this? He never figured it out. He came away like, I don't know. And the Carnegie Institution actually gave Luther Burbank $10,000 a year so that they could send someone, a geneticist, to go and get the magic. And he tried following Burbank around. Burbank, get away from me, kid, you bother me, for a year and a half. And this scientist, George Schull, just wrote back and said, there's no science here. And they just gave up. Ironically, Schull then went off and invented hybrid corn.
But don't you actually say, I can't remember exactly, but don't you actually say that DeVries discovered that Luther Burbank had no theory of heredity?
Yeah, well, Schull was the one who DeVries was I guess I haven't figured this one out. And he kind of went away, and he said nice things about him, because he admired him as a plant breeder. I mean, everyone admired him, but when Schull really was on a mission to write science about it, he said, "This is ridiculous. This is bogus." And Burbank kind of was sad that towards the end of his life, he got accused of fraud, and and he was selling plants that maybe weren't living up to his promises, and but he had this iconic, even after death, he had this iconic place. I mean, Frida Kahlo did a painting of him after he died. He's an amazing person.
I want to follow up, okay? But first, I forgot to do what you did for me, which
you.
is hold up the book. They tell you if you're on a talk show, hold up the book. She has her mother's laugh, Carl's massive and majestic book is what we're talking about. 2/3 of the way through, I hit a point where my jaw dropped, and I was very interested, and I was learning a lot, but there was a point where I started saying, "What? No way!" And by the way, if you happen to ever see the New York Review of Books, there's a review in the current issue by Tim Flannery, an important, interesting Australian scientist, review of this book, Carl's book, and Tim Flannery reacted much the same way. He got to these two subjects, and he said, "What?" And he talks about them in the review. Mosaicism and chimerism. So that's really the jaw-dropping where it starts for me. Tell us a little bit about creatures as mosaics, and then a bit more about creatures as chimera.
Well, I think these are in the same category as horizontal gene transfer, where if you hear about it at first, you're like, well, that couldn't happen. And then you say, well, okay, if that does happen, that can almost never happen. And then you discover that it's actually pretty common.
Yeah.
And it breaks these rules that we have in our heads. So mosaicism, I guess for me, these two things, mosaicism and chimerism, they challenge your idea that we have, that you are your DNA, in the sense that you started as a fertilized egg with a set of DNA, totally unique. That's what sets you apart from everyone else, and that is, in every cell of your body. That's how we define you. You steal something, and we get some skin samples, and we sequence your DNA. That's you, and we can arrest you because that's you. But the fact is that, for all of us, as soon as the fertilized egg starts dividing, there's a chance that the DNA will mutate again in a cell, and again and again and again, so that a cell in your eye and a cell on your big toe probably have different mutations because they have a different history through your development. And so we can draw a tree, another one of these trees, a tree of your cells. And August Weismann actually would draw cell trees, and they're different. And it's not just that they're different organs, but they actually have a different sequence of DNA in them. And this could be incredibly important in all sorts of ways. But in one way is that a lot of diseases may be the result of mosaicism. So that in effect, you could be as a fertilized egg, you might not have a hereditary disease, but then along the way, a mutation shows up, and let's say that it affects your muscles, and you have this muscle disease. But if I took a blood sample and looked for that mutation, I wouldn't find it because it's not in your blood. So you are a mosaic of different genomes. So we're all mosaics. That much is clear. But there's another way that we can become mixtures, which is that some of us are chimeras. And a chimera is any organism that's made up of cells from more than one individual. And this started to come to light with some really strange cases. For example, a woman in the 1950s gave blood, and they found she had 2 blood types. And they said, no, that would mean, no, there's a mistake. Someone mixed blood from 2 people together. Actually, what happened was that this woman had a twin brother. He died when he was young. In utero, their embryos had traded cells. Those cells, being fetal cells, had taken hold in each other's bodies and had diversified into blood, into bone, and so on. So they were mixtures of each other. There are mothers who have almost lost custody of their own children that they gave birth to because genetic tests indicated they couldn't possibly be their children. Because, again, testing from the blood and the eggs that gave rise to the children came from different people.
This is so counterintuitive. Back up a little bit and tell them about free martins.
Right, so free martins actually were sort of like, free martins are cows who are the cow version of what I just told you. In other words, they're twins, but the females which would turn out to not be great for giving milk. And when anatomists looked at them, they discovered they had some male traits in them. So they're a chimera of a female calf and a male brother calf. And so people didn't know. They just were like, well, this is strange. They identified them in the 1700s as being anatomically odd. Didn't know what to make of them. Turns out they're chimeras. People can actually be chimeras, where you have different, male and female combined.
Fraternal twins?
Yep. And same twins and so on. Women have an additional opportunity to become chimeras if they become mothers, because the fetus is shedding cells. A woman's immune system will destroy most of them, but sometimes they don't. And a woman gives birth, the fetal cells are hanging around, and they just wander through the body, and they become whatever's around them.
Some of them are stem cells.
Well, they all have this sort of stem cell quality to them, and which means that they become muscle cells. They can become skin cells. Yeah. They seem it's a weird and mysterious thing. It seems that if a woman who has these fetal cells in her body, it's called microchimerism. If she gets a cut and she starts to heal, it's the fetal cells that seem to really get attracted to the wound, and they help to heal it. So you find a lot of fetal cells around a wound. There was a study where scientists said, okay, let's find a lot of cadavers of women who gave their bodies to science who happened to be mothers of sons. And they cut open their brains, and they looked inside, and they found neurons with Y chromosomes in there. These are most likely their sons' neurons. I mean, the fetal cells went into their brain and said, hi there, we're here to help out, and they became neurons. Functional neurons. And this was, I forget the exact number, it was something like 20% of the women. Yeah. So these are very crude ways of getting at it. Those are probably a gross underestimate. So probably it's, I'm going to guess maybe a third of mothers are chimeras, but the numbers are super rough. If it turns out to be more, it won't surprise me one bit.
So this, among other things, raises the same question that people ask me when they hear about horizontal gene transfer, endosymbiosis, and some other things. What does this do to the concept of a human individual as individual?
Right. I think human, when we talk about human individuals, I think it's like the psychological shorthand we use to make our way through the world and to deal with these, bags of cells that kind of look like us. That's it. I mean, like, there's nothing deep and fundamental about our individuality. There just isn't. I mean, the mothers among you, a number of you are chimeras, and then a few percent probably of the rest of you are also chimeras with a twin you never knew you had, who probably died in the womb. Like, you're a composite, and all of us are mosaics. There's no one magic code that defines who we are. So individuality is way overrated, and even what a species is overrated.
Yeah.
So you've got horizontal gene transfer kind of mushing together genes from bacteria that have been separated by billions of years. I mean, we've got DNA from Neanderthals and Denisovans and viruses and all sorts of stuff. It's a hodgepodge in there. So, and whenever I'm writing about Neanderthals, someone will say like, well, are they a separate species or not? And then I'll say, well, look, I'm a journalist. I'll call the scientists and say, people want to know, is that are Neanderthals a separate species or not? And then all the scientists say, please don't ask me that question.
How long is a piece of string?
Yeah, right. Where is the divide between two mountains? I mean, it's just it's arbitrary in these cases. So, yeah, and it's fun to be just in writing about science that you end up having to deal with philosophy. So, yeah. Well, we should I'd like to sort of jump back maybe. We talked about you as an English major at a certain place, and how do you go from being an English major to someone who is getting into the finer details of mitochondria?
Yeah, I'm still trying to figure that out. I did my undergraduate and my graduate work mostly in literature, obsessed with William Faulkner. And then I went off to Montana in 1973. I had already published and I wanted to be a fiction writer. And published one novel. Was very lucky and very young. Right out of college, I published my first novel. 49 years ago. Yeah. Well, I don't know. No, more than that. It's more than that, actually. Anyway, I'm not good at mental math. And then I paid my dues as a writer. You got to pay your dues sometime. I paid my dues not before the first book, but between the first book and the second book. So I spent 13 years or so in Montana doing those menial jobs that a struggling young writer does. Tending bar, waiting tables, working as a fishing guide, etc. And gradually discovered, besides discovering that I couldn't make a living as a novelist, I discovered that I really loved nonfiction about science, especially nonfiction that was imaginative and artful. And when I say imaginative, I don't mean making stuff up. I mean excellent writers who used fact to create mosaics, artful mosaics, storytelling. I started reading Haldane, I started reading early John McPhee, reading early Stephen Jay Gould, a number of others, and I discovered that nonfiction could be an artistic branch of writing. And so I started trying to do it and figured out by trial and error how to be a magazine writer, and then finally said, well, I want to do a big, ambitious book. I read a little bit of Ed Wilson and Robert MacArthur about the theory of island biogeography. I read very early, and I started writing this book about island biogeography. I read very early on a book called The Secular Arc, which I recommend to you all. It's a book about the history of biogeography by Janet Browne. I think it might have been your PhD thesis, yes? Okay. Wonderful book, one of the first things I read. And I thought, what a wonderful field, the study of why different creatures live where. And then I spent 8 years on that book, and when I was finished coming out of that rabbit hole, I guess I was a nonfiction author.
Mm-hmm. How do you, you write about, you go places and see living things. That is such an important part of what you do. Like, and I just sort of wonder, like, how it must be. I don't know. It just must. When I'm reading your stuff, I'm like, how do you like take notes while you're like, trying to not get run over by an elephant, or it's day 30 of this hike across the Central Africa, and you've got your feet wrapped up in packing tape or whatever it was.
Duct tape.
Duct tape. Yeah. I mean, like, I would think it'd be hard to think of the craft of writing when you're in the elements like that.
The answer to that question is a ballpoint pen, a small notebook, and a Ziploc bag.
All right.
Very low tech. And the things that Carl was alluding to, I've done a lot of them for National Geographic magazine, therefore in partnership with some of the world's best and toughest photographers. And I always feel sort of smug and pitying toward the photographers I work with. Sometimes we're in the field together, sometimes not. And we're at the airport in Brazzaville, Congo, or somewhere, and they're trying to get their 13 Pelican cases of expensive equipment through customs and be sure they can get it back out again. And one of the cases is missing in Paris, and it's got this V lens in it, and I've got this notebook and Ziploc bag and a ballpoint pen. So that's the answer to that.
It's good to travel light.
I read, when I learn something new in science, a breaking event in science, it would be nice if we were arguing with each other, but we're not going to argue with each other tonight. We're pals, and we have high mutual regard, and we probably disagree on some things that we could argue about. But I open the New York Times online, and I see Carl's got an article about this or that, about 8-lettered DNA, 8-lettered artificial DNA, that was last week or earlier this week, or about the latest discovery from do you say Denisovans or Denisovans?
I think it's Denisovans.
Denisovans, okay. The latest discovery from Denisova cave in south central Siberia. And that's how I find out about these things. He does a number of different kinds of writing, but one of the things that he does that I have high respect for, I depend on, and I don't ever want to do, is he gets journal articles that haven't been published yet, and he breaks that news to the world through the New York Times. This is what is going to be published in Nature tomorrow. And you do that with a computer and a telephone, right?
Yeah, I mean, I get wind of papers coming out, or the journals sometimes set things up with embargoes. But what you do is you just you can do. I will read the paper, which is usually kind of a jumble and sort of hard to understand because it's not well put together, and then.
To put it politely.
All right, all the scientists groaning, take a writing class, okay? You probably didn't teach yourself statistics, okay? You don't have to teach yourself writing. I mean, it's okay. And then I'll read papers by other people, which again, are a bit of a struggle. And then the key is that for me, if I'm doing these articles on a weekly basis, is the phone call. And I'll just sort of, I will just say, all right, let's just start at the beginning, and I'm going to stop you, and I'm going to say wait, you just skipped a step. What are you talking about? And they'll be like, oh, yeah, right, I should have told you this. Oh, right. And eventually you get people, I just find when people are talking, they tell stories about their lives, in this case, their scientific research, in a much more compelling, clear, concise way, often with quite beautiful metaphors. They'll just boom, all of a sudden they say something and they're like, ooh, I've got to make sure I'm recording that because that is going in. That's a quote that just sums up exactly what they're getting.
Are you recording these phone calls, did you
Always,
say?
yes.
Yeah, always.
Yes. The Skype record plugin is my savior.
You didn't take shorthand at Yale?
Did they teach shorthand at Yale?
No.
I missed that one. No, I'm not great at that. No, I have to admit that I I need the prop of
I'm
recording.
so glad to hear that.
And now we're sort of reaching that age where computers can start to transcribe these things, and so I'm like in the promised land.
Did you ever, I know Ed Young had this experience, did you ever have occasion to call up Carl Woese and ask for a reaction?
Yeah.
How did that go?
Not well. I mean, yeah. I mean, I was at Discover. Gosh, it's so long ago. I can't really remember what I was even calling him about. Maybe I was reporting a short piece or something, or checking something, and I thought I knew I had a vague idea of his importance, but he was just like he just suddenly just started insulting everyone, and like it was totally tangential, but he wanted to just start cutting people down, like, and wanted me to hear it, and I'm like, I'm just a young reporter, and I don't know what to do with all this.
He was crotchety.
He was
notoriously crotchety, and I think insecure as well as brilliant and accomplished. Yeah.
We've got a few minutes left. I mean, maybe we should before we open up things for questions. I mean, why don't we sort of like talk about our different sort of perspectives on like what's happening these days that. Aside from the things we've talked about, that's really exciting us sort of in the world of science. So,
is Lamarck back?
Is Lamarck back? Why can't Lamarck rest in peace? I mean, come on.
I read it in your book.
I know.
It's the question. I'm not saying you answered it, but the question is in there, right? You're asking yourself, is Lamarckism, and we should stipulate by Lamarckism, shorthand, we mean the one particular idea, the inheritance of acquired adaptations.
Well, right. That's what Lamarck said. Yeah, and I think that's the problem, is that people don't really appreciate what Lamarck was trying to say. Lamarck was really like talking about a theory of evolution, and he deserves incredible credit for that. He was interested in adaptations. He wasn't interested in like, if you raise a mouse in horrible stressful conditions, are the mouse pups going to be messed up? Like that. That's Lamarck would be like, really? Like,
or if you cut off their tails.
Yeah, well, exactly. The classic experiment to show that Lamarckism wasn't working. Lamarck is being resurrected all the time, and I think we journalists are to blame a lot for that. Although scientists sometimes will put Lamarck on their journal covers, I think we need to sort of cool down a bit. I mean, and if you're a plant, which I'm not. I would say that certainly the inheritance of acquired traits is a thing. I mean, epigenetics really does seem to be important to plants, and it's a way for them to survive and to thrive in a changing environment. So if there's a drought, descendants of a plant will be better prepared in a way for a drought for several generations.
Yeah.
But people have wanted to jump to explain all things about human life as well with that.
Okay.
And I think it's way too hasty.
There's a lot in here about epigenetics, which is fascinating. And that's what brings you, I think that's the main thing anyway, that brings you to the question of, is Lamarckism back? Is it real? Is it viable under some circumstances? But the reason I get asked it is because people wonder, well, horizontal gene transfer, endogenous retroviruses, retroviral infections that infect the germline of humans. 8%, one of the things I talk about at the end of my book is that 8% of the human genome is viral DNA that has been acquired over the evolution of mammals and particularly of primates because of retroviruses inserting themselves into human germline cells, into eggs, sperms, or the stem cells that make them, and therefore becoming heritable. Viral DNA, including a stretch of viral DNA that is crucial to the formation of a layer in humans between the placenta and the fetus. So that human pregnancy is not possible without this stretch of DNA that was originally a viral envelope gene and is now creating this layer with a long fancy name. Is that the inheritance of acquired characteristics?
Well, it's, what Lamarck was talking about was an experience in the environment, interacting in the environment that changes an organism, and then that change gets passed down. But certainly, like, it's a kind of infectious heredity, certainly.
Infective heredity.
Actually, ironically, maybe the best example, I think, of this is, could be CRISPR. So people think about CRISPR as a way to edit genes, which it is, but we stole it. We stole it from bacteria. They invented it through evolution. It's a way that they can defend themselves against viruses. They can they can recognize bits of viral DNA and cut it, disabling the viruses.
And they recognize it because they've been infected by that virus before, right?
Right. So if they can survive an infection, that gives them a chance to size up this viral DNA so that the next time that they get infected, they can use CRISPR to shred it. Then, what bacteria do to reproduce is they just divide, and they just, and they actually, each of those new daughter microbes is going to inherit that memory, that experience of being exposed to that virus. They have Their genes have been altered so that now that they are ready to fight against that virus should they be infected by it. And so a scientist named Eugene Koonin has argued that CRISPR is Lamarck. And a lot of people don't agree with him, but he's sticking to his guns. And it's an interesting point.
Getting to visit people like Eugene Koonin, are we wrapping it up, is one of the reasons why he and I have a great job.
Yes.
Both spent time with this wonderfully crazed Russian-American man named Eugene Koonin at the National Library of Science in Bethesda, right?
National Library of Medicine, yeah.
National Library of Medicine, yeah.
He's got a million ideas in his head, and half of them at least are fantastic.
Yeah.
So why don't we can take some questions.
Let's a round of applause.
Okay.
Thank you. Thank you.